Morning, children. Today we are going to discuss about the biomolecules chapter, lipids and proteins and polysaccharides information. So you know very well the molecules we discussed previous classes, like carbohydrates, proteins and lipids are there, and what is the major difference between what you called essential amino acids, non-essential amino acids, okay, and saturated fatty acids, unsaturated fatty acids, monosaccharides, disaccharides examples, polysaccharides examples, like that in the previous class we studied. So now today we are going to discuss about the complete information about proteins. Okay, so before going to the proteins, some important points are there. That one once we will recollect, then we will go to the proteins. Okay, children, observe very carefully. These all are very important points in the NEET exam point of view. Okay, the first thing is I am telling about the lipids. So before first, what is biomolecules? The molecules which are present in the life, like what you call carbohydrates, protein, all organic compounds and elements. Okay, especially organic compounds, carbohydrates, proteins, lipids, okay, fatty acids. These all are biomolecules, and elements like hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, calcium. These all are nothing but what elements. They are present in the living matter and even non-living matter also. So the first why how to analyze the composition of the living tissue. For example, take one liver piece and grind it in the trichloroacetic acid and filter it by using which cloth? Cheesecloth or cotton. Okay, then we will see insoluble acid insoluble fraction is there. Acid soluble. Acid soluble means micro. Acid insoluble means macro. Like that, the macromolecules will be there. Okay, all the carbon compounds that we get from the living tissues can be called biomolecules. Very important definition. All the carbon compounds which we are getting from the life is called biomolecules. So for example, if you want okay, dry inorganic elements you want to get, in the sense, first what we have to do, take one tissue and leaf like that and burn it and make ash and identify that composition of that particular ash leaf. So you will see same what you called carbon, hydrogen, what you call nitrogen, sulfur, some elements will be, okay, what you called calcium, magnesium, sulfate, phosphate are also seen like that. So there will be living and non-living, both have the same components, but the composition, the percentage is different, and the arrangement of molecules, arrangement of elements, arrangement of compounds are different in the living form. So there will be different types of proteins are there, carbohydrates, lipids are there. So what are the amino acids? You know very well, amino acids means what? The proteins means made with amino acids, polymer of amino acids. So 20 types of amino acids, right? So acid group, amino group, and the same carbon is there in this alpha carbon, alpha amino acids. Like that, amino means amino group, NH. Carboxyl group, COH group, same carbon in the sense alpha amino acids. Are you following or not? So like that, some are acidic, some are what you called basic. Okay, based upon the R group, the amino acids are will be there like that. Okay, children, that is the most important amino acids are there. Acidic amino acids and basic amino acids. So we are going to discuss about the Zwitter ion. What is Zwitter ion? Both plus minus. What is called plus minus on the same amino acid is there in the sense that is called Zwitter ion. Okay, Zwitter ion. So here I am telling some amino acids, examples of acidic amino acid. Acidic amino acid example, what? Amino acid, glutamic acid. Okay, like that. And a neutral valine. Okay, basic lysine, glutamic acid, lysine, valine, like that acidic, basic, neutral amino acids are there. So like that. So what we are doing first, taking one tissue, disturbing lipids. Lipids, one important question. Lipids are macromolecule or micromolecule? The question is, lipids are not strictly macromolecules. Why? Why? Because lipids will be actually soluble. We will see in the filter it. But why we are considering macromolecule? Because see now, the grinding tissue, at the time of grinding the tissue in the acidic trichloroacetic acid, disturbing the cell membrane. Cell membrane contains lipid. This broken into pieces, not water soluble. So those all pieces in the form of vesicles get separated along with acid insoluble. So they will attach to the what you call other macromolecules and insoluble. So that's why the lipids are not strictly macromolecules. Actually, they are micromolecules. Because of their insoluble property, they will be outside of the acid insoluble composition. Okay, so this is a very important need bit. That's why especially I wrote lipids are not strictly macromolecules. Why? When you are grinding tissue, the disturbing the cell membrane, broken into pieces, and they are not water soluble, they will be form of vesicles, get separated along with acid insoluble pool. So this is about what you call lipid. And protein, what is it? Proteins, polymer of amino acids. Proteins, whenever the word you should hear, this is a polymer of amino acids. 20 types of amino acids. Heteropolymer means what? One is for example, valine, like this only there in the sense. This is homopolymer. Instead of that, okay, leucine, valine, histidine, okay, and methionine, like this different amino acids are joined together in the sense, that is called heteropolymer. This will be considered as one protein, heteropolymer protein. So like that, there are homopolymer proteins, heteropolymers, but maximum heteropolymer proteins are there. So there are essential amino acids, non-essential amino acids. When we will call the essential amino acids, that is important. When we are not getting the food, only body only preparing, in the sense they are non-essential. Simply I am telling, through the body, our body itself produce some amino acids, they will be called non-essential amino acids. And through the food, which amino acids we are getting, those are called essential amino acids. Very simple. Through the food we are getting, in the sense they are essential. Our body itself preparing, in the sense they will be called non-essential. So protein, the most abundant protein on this animal world is collagen. What is called? Very important need bit. The most abundant animal protein in the animal world is collagen. In the plant world is most important, that is called rubisco. Very important. These two important enzyme. So most important abundant animal protein is collagen. Very neat, important need bit. And most important what is called plant protein is what is called rubisco. What do you call that is rubisco. So like this, there will be a different important concepts are there in this chapter. Okay, children. Next, polysaccharides. Yesterday class we discussed. What are the polysaccharides? Yesterday we discussed or not? So polysaccharides means more carbohydrates. What do you call? More carbohydrates, glucose molecules, more in the sense, for example, cellulose, more amount of glucose, glycogen, animal stored starch. So here I am telling, cellulose is a, you know very well, it is in the plants, cell wall it is present. More amount of glucose molecules togetherly form the cellulose. Glycogen means what you called animal, what you call stored fat, stored carbohydrate. More amount of glucose will be there. So polysaccharides are very useful. Why? Because cellulose, cell wall, storage, carbohydrates, energy sources for us, main energy source. So here you should have to get to one doubt. When the starch react with iodine, blue color will come. You know that is the indication for photosynthesis experiments. We know that that's a major principle. Okay, how we can identify the carbon dioxide is essential for photosynthesis. How can you identify the light is essential for photosynthesis? Okay, for such kind of the reactions, we will do the test with the iodine, with that chlorophyll. Then starch formed, in the sense blue color will be formed. That is different. So when the starch react with iodine, blue color will come. Cellulose also carbohydrate, polysaccharide, but when the cellulose react with iodine, no blue color. Why? That is the question. Why? Because in the cellulose, no complex helices. Means no complex foldings. Helices are not there. Hence, iodine cannot hold the, what you call, hold by the, that one. So, cannot hold by the cellulose. That's why cellulose, even the react with iodine, there is no blue color. That's important bit. Then amino sugars, chemically modified sugars, okay, like this aniazine, N-acetyl glucosamine, these are also related to which one? The polysaccharides, N-acetyl glucosamine, chemically modified sugars, N-acetyl glucosamine, these all are nothing but what? The amino sugars, okay, so that is important. Next come to nucleic acids. The nucleic acids, you know very well, that is life for us. Characters, everything will be in the nucleic acids, right? So, first of all, we are discussing what are the nucleic acids and how many types will be discussed. The other type of macromolecule is nucleic acid. That one would find in the acid insoluble fraction. So, it will be any tissue of the nucleic acid. These are polynucleotides. So, number of nucleotides are there. So, polynucleotides, okay. These are comprised with the true macromolecular fraction of any living tissue of cell. Nucleic acids are the building blocks. Building block is a nucleotide. So, because of nucleotides, nucleic acids are forming. That is important. So, what is a nucleotide? Nucleotide means you know very well. Nitrogen base, phosphate, and what do you call? Nitrogen base, phosphate, and sugar. This together called the nucleotide. What is called? Nitrogen base plus sugar plus phosphate is together called nucleotide. Only sugar and nitrogen base means nucleotide. Phosphorus is there, phosphoric acid is there, nucleotide. So nucleic acids are made with nucleotides. What are the nitrogen bases? So number of nucleotides forms the nucleic acids. What are the major important nucleic acids? Two, DNA and RNA. Deoxyribose nucleic. So without one oxygen. So second position there is no oxygen. So that's a deoxyribose nucleic acid. So there. What are the nitrogen bases here? What are the nitrogen bases? Adenine, guanine, cytosine, thymine, uracil. These are divided into purines and pyrimidines. What are the purines? Adenine, guanine, purines. Cytosine, thymine, uracil are pyrimidines. So the sugar which is present in the ribose sugar we are discussing or not? That is nothing but what? The ribose sugar nothing but pentose sugar. D means deoxyribose, like that there will be structures are there. Next come to the proteins. Major discussion about the proteins. Till now we discussed lipid, protein, what you call major macromolecules. Now in depth we will go one by one. First one, proteins. What is a protein? Protein is mentioned earlier as a heteropolymer containing strings of amino acids. Primary structure, strings of amino acids. Okay, this is important. The structure of molecule means different things in the different contexts. In organic chemistry, there will be, you know very well, formulas are there. So, how can you say the amino acid means? Just like this 1, 2, 3, 4 amino acids together like a peptide bonds. Okay, this amino acid means that is called protein. So, linear. The sequence of amino acids, the positional information protein, which is the first amino acid, which is second, and so on. So, first, second, like that is there in the sense that kind of structure of protein is called primary structure. It is called primary structure. So, like that, the proteins are there. The first amino acid also called N-terminal amino acid, and last amino acid called C-terminal. N-terminal, C-terminal. First amino acid is N-terminal. Last is C-terminal amino acids are there. So they are rod-like. They are rod-like first linear primary structure. But foldings are there in the sense right hand helices, other regions of portion, threads are folded into other form is called, see, like this. This kind of foldings are there in the sense that is called secondary structure. Nothing but proteins or amino acids only. Like this, it's the amino acid. So next one, the tertiary structure. Tertiary structure means hollow, what is called woolen ball. See, hollow woolen ball like that. So all foldings, three-dimensional view, hollow woolen ball. That is a tertiary structure. This tertiary structure is the most the tertiary structure is the most useful in the all metabolic activities, means enzymes. Best important thing is that form example is enzyme. See, enzymes will be bind with the substrate, right? Because of this form only. So the product will become. So like that, the tertiary structure, hollow woolen ball, there is a three-dimensional view is there. So many biology in the three-dimensional view, the tertiary structure, all amino acids will be arranged with peptide bond like woolen, hollow woolen ball. So in the many biological activities, the tertiary structure is very important and performs the enzymatic actions, increase the rate of reactions, produce the products, and so many metabolic functions they will be performed. Next one, the quaternary structure. What do you call quaternary structure? The quaternary structure is cube-like structure or plate-like structure. So quaternary structure, for example, you know very well, hemoglobin. Hemoglobin consists of 4 subunits. So two of these are identical to each other. Hence, 2 subunits of alpha type and 2 subunits. So quaternary means in the name itself, answer is there 4 subunits. 2 alpha groups, 2 beta groups, like hemoglobin structure. So that is called, what do you call, that kind of structure is called quaternary structure. What do you call quaternary structure? So like this, the proteins are available in the which one? Primary structure, linear, like a rod. Secondary structure, like foldings. Tertiary structure, like hollow woolen. Quaternary structure, like a 4, what do you call, cube or plate like. Cube or plate like structures are there. So these all are the importance for the protein information, important protein information. So next important thing is, next class we will be discuss about what is called nature of bond linking monomers and polymers. What bonds are there between the peptide bonds, means what? Okay, glycosidic bonds, diester bonds. Okay, so such kind of bonds, how these bonds are joining together and forming the molecules, that concept will be discussed in the tomorrow class. But up to that, this hemoglobin, you know very well. What is hemoglobin? Quaternary structure. What is the major element present in this hemoglobin? That is iron. So for four structures, you know, for one 4 molecules of oxygen, it will be bind to form the oxyhemoglobin. So you know that information in the respiratory, what you call circulation system, you might, and respiratory system, you might we know that. So 4 compounds, 4 molecules are there. So that's why it is called quaternary structure. So what are the need bits? Most essential, most abundant, what you called protein in the animal world, that is called collagen. In the plant world, rubisco. Protein means polymer of amino acids. Nearly is a heteropolymer. So different types of amino acids. Why we are calling essential amino acids? Because we are getting through the food. Non-essential, why we are calling? In our body itself, the production is going on. That's why they will be called non-essential. So polysaccharides. What are the polysaccharides? Cellulose, glucose. Okay, group of what you call carbohydrates, means simple glucose molecules togetherly form complex like polysaccharides like cellulose, glycogen. Why iodine react with the starch and making the blue? Because they have helices. Why cellulose not producing the blue color when react with iodine? Why? Because no helices. Okay, so chemically modified sugars, examples what? N-acetyl glucosamine, NAG and glucosamine, they are the part in the bacterial cell wall. So next, what is the what is called pentose sugars present in the DNA and RNA? In RNA ribose, in the DNA deoxyribose. So O group will not be present in the deoxyribose in the second position. So proteins, examples for acidic amino acid, glutamic acid. Example for what is called basic amino acid. Okay, what is the basic amino acid example? Histidine. Okay, neutral. Okay, histidine is there or not? So like that, there will be basic amino acid and acidic amino acids will be there in our structure also. So these are the major important concepts about our syllabus. Okay, So first amino acid. What is that one? First amino acid carbon. What is the one end is amino acid. N terminal. Another end is carboxylic terminal. First end is N terminal. Second one is carboxylic terminal. Also will be there. Like that, there will be different concepts are there to discuss in this chapter. Okay. In the next class, we will be discuss about what you called the nucleic acids and what you called the deep depth about what you called the bonds and peptides, carbohydrates, like this we will be discussing in the next class. So valine, histidine, glutamic acid, like that acid, bases, and this kind of things will be there. Acid amino acids, acidic amino acids, basic amino acids, and what you call neutral amino acids also will be there according to the position of the R, right? So next, another important thing is in the bonds, whatever the bonds are there, those bonds will be discussed in the coming class. What are the bonds? Peptide bond means what? Glycosidic bond means what? What you called is ester bond means what? What is ester bond? How they will be present in the in between which kind of compounds ester bonds are there? Like that will be okay. So what is the example for neutral amino acid? Valine. Okay. Acidic, glutamic. Basic, lysine. Basic amino acid is lysine. Valine. Valine for neutral. Acidic. Okay. Glutamic acid, like that. There will be examples also there in amino acids. So thank you children. Next class we will be discuss about bonds.